Hi, I'm Sandra Arismendez. I'm the Water Quality Monitoring Coordinator at the Meadows Center for Water and the Environment. We're out here today studying the E. coli bacteria and optical brighteners on Cypress Creek. We're looking for potential contamination from on-site septic systems from the residential area surrounding the Wimberley Valley area, and also looking at the bat colony, the impact of the bat colony on E. coli bacteria on the water quality in Cypress Creek.
Hello, I'm Daniel Vasquez. I'm a student research assistant with Texas Stream Team at the Meadows Center. And along with our E. coli bacteria study that we're doing, we're also having a tampling study. And the tampling study is used to detect optical brighteners in the water. And optical brighteners are typically found within toilet paper, tissue, soap, detergents. So we're suspecting that if we're able to detect optical brighteners within the water, that there could be potential septic system leakage or something malfunctioning. So along with our E. coli water samples that we're grabbing today, we're also taking the core measurements with our probe kit. And the probe kit consists of a dissolved oxygen meter and a pH and conductivity meter. Additionally, we test for air temperature, total depth, and water temperature.
Part of the project is to collect a water sample and analyze it for E. coli bacteria. So we use these sterile Whirlpack bags, attach them to our sampling pole here, and then reach out to the center, as far out towards the center of the stream to collect the water sample. So the sample is then taken back to the lab. It's plated using the ColiScan Easy Gel media and Petri dish. It is then placed in an incubator for 28 hours. And then the E. coli bacteria colonies that form on the Petri dish are counted and documented. So we're about to retrieve our tampling product for our 3-day deployments. Today we're retrieving tampling material from bottle A. So it's been deployed for 3 days. I'm gonna rinse it in the creek water. Place the foil. I'm gonna squeeze out all the excess water. Seal that to prevent any UV light from entering and degrading the optical brighteners. Place it in a baggie. Purpose to redeploy the tampon. There it is, suspended in the column. We've got a couple of rocks down at the bottom to weight it down. Okay, so each of the 3 bottles have slits at 1-inch increments all the way around. So that allows the water to flow through the bottle and come in contact with the organic tampon, which is then designed to absorb the optical brighteners in the water. We've got all 3 bottles attached to this 20-pound weight, and so we will redeploy in the creek.
Hi, I'm Desiree, and I'm one of the water quality monitors with the Cypress Creek Tampling project. Right now I'm going to explain to you how we detect our optical brighteners. So after we retrieve the tampon from the water sample, we make sure that we keep it in a dark environment by wrapping it in foil, and it's in a dark environment until it's in the lab analyzed. We use a blacklight UV lamp to shine on the tampons, and if it fluoresces, then the result is there is OBs, optical brighteners, on the tampons, and it's going to be a bright blue fluorescent light. And in some cases, we are getting some red fluorescence.
After we gather our samples and we're going along with the plating process, we want to make sure that we're incubating our samples around 33 degrees Celsius for roughly 28 to 31 hours. And we're going to make sure that we're counting the colonies with a distinct dark blue and dark purple center. Each one of these dots will represent 1 colony towards our total E. coli colony count.